Today I will talk about a long-noncoding RNA that we have identified several years ago in the lab, and is one of the first long-noncoding RNA that have been described in human cells. And so the story started because in our laboratory we were interested in this particular HRNP protein, and HRNP proteins are proteins involved in RNA metabolism, different aspects of RNA metabolism. And the idea was just to find a novel molecular partner of HRNPA1. And we perform a two hybrid screening in yeast to look for partner of this particular protein, and we pick up that protein that belongs also to the HRNP protein family, in the sense that what you can see here is the whole set of HRNP protein that can be co-immunoprecipitated by a single antibody, for instance, an antibody against HRNPA1 or HRNPA-C, when you preserve an RNA integrity. So all these proteins are bound to RNA, and they are co-immunoprecipitated by all together. So in order to understand if this protein, HRNP-up, can actually interact by itself with HRNPA1, we decided to explore different cell growth conditions, and one of these was just to test the effect of heat shock on the subcellular distribution of this protein. So this is the distribution of the protein at 37 degrees. This is a cell nucleus. You can see that it is distributed throughout the cell nucleus, and you can see also some site accumulation of accumulation close to the nucleoli. But when you heat shock the cells for 1 hour, 42 degrees, and then you allow the cell to recover 3 hours, 37 degrees, the distribution clearly changes. And this is, again, is a cell nucleus. You cannot see the nucleus just because all the protein accumulate in specific dots. And you can see, usually in HeLa cells, you have 5, 7 dots per cell. In other cells, you have 1 or 2, depending on the cell type, and probably also on the ploidy of the cells. So we look also at the electron microscopy, what these structures are. And in HeLa cells, after its shock, you can detect these large bodies. Just to give you an idea of how large are they, these are ribosomes in the cytoplasm. So they are huge structure, and they are due to the assembly of these subcomponents, this structure here, that accumulates and eventually give rise to these large bodies. And these structures are, because of their shape and a number of features, they can be recognized as perinuclear granules, perichromatin granules. And perichromatin granules are structure that are actually ribonucleoprotein particles, highly packed. So you have highly packed ribonucleoprotein particles that are synthesized, and they collapse to form these large structures. So since they are ribonucleoprotein particles, it's not surprising that it is not only HAP that is present in these structures. You can see there are several other SR proteins, SRSF9, 7, SRSF1, but not all SR proteins are present there. So for instance, SRSF2, that was previously called SC35, is not present in these structures. But surprisingly, another group in France describes something similar, and they were interested in studying heat shock factor 1. So if you look at heat shock factor 1, also heat shock factor 1 accumulates in nuclear structure after its shock, and you can co-stain the cells with antibody against the heat shock factor 1 and the antibody against HAP, And what you can see is that after 30 minutes at 42 degrees, ICGEB factor 1 is already present in granules, whereas HAP is not. And then after 45 minutes and 1 hour, they colocalize perfectly in the cells. But then when you have the recovery from its shock, ICGEB factor 1 redistribute throughout the cell volume, whereas HRNP up is still in granules. So there is different proteins. Some proteins are involved in transcription, such as ICGEB factor 1, ICGEB factor 2, RNA polymerase 2. They accumulate in this structure. But they and on the other side, also protein involved in RNA metabolism, such as HRNP up, HRP-M, some splicing factor of the SR family, or SAM68, that is another RNA binding protein. They accumulate in this structure. So these bodies have a dual nature. On one side, they are involved in transcription. The other side, they are involved in pre-mRNA processing. And the kinetics of recruitment and release from the body is different, because these factors that are involved in transcription are recruited first, and protein involved in RNA metabolism are recruited after. So we decided, we tried a lot to understand what is the meaning, and not only the meaning, what is the mechanism for the formation of this structure. And we took advantage of one observation, and the observation is that nuclear stress bodies, that we call the name that we gave to these particular structures, are specific of human cells. So if you try if you eat shock hamster cells or mouse cells, these bodies do not form, but they form very, very rapidly in HeLa cells. So you can exploit this difference, this specificity for human cells, and you can use human-hamster cell hybrids. So there are cells that contain 1, 2, or a number of, they are hamster cells, but they contain a number of human chromosomes. And for instance, only a single human chromosome. And you can see here, for instance, if you take a hamster cell containing chromosome 14, nuclear stress body do not form. If they contain chromosome 15, nuclear stress body form. So there is a set of human chromosomes that are sufficient for the formation of stress body in hamster cells. So there is some peculiarity of these chromosomes that is sufficient for the formation. So the cell can form, but it lacks something, and this something is provided by several human chromosomes, not a specific human chromosome, that in a sense argue against the presence of a protein coding component on the chromosome, because otherwise it's difficult to imagine that the same component is present on different human chromosomes. In any case, this is the analysis, electron microscopy analysis of nuclear stress body in HeLa cells and in hamster cells containing human chromosome 15. And we focus particularly on chromosome 9, human chromosome 9. And we use, this is just because we had different hybrids that contain different portion of human chromosome 9, the entire chromosome, or the entire long arm, or the entire short arm, or a region spanning the centromere. And you can see that all these fragments of human chromosome 9 were sufficient to trigger the formation of stress body after its shock. And they share this region. And in this region, you have a small portion of the short arm of chromosome 15, chromosome 9. You have the centromere, and this very long band that is heterochromatin. It's composed of is about 15 megabases of satellite III DNA. So a repetitive DNA element typical of human chromosome, that is a satellite III DNA. And this region is composed mainly of this, by this sequence. So the idea was, is it possible that this sequence actually is the region on which satellite III nuclear stress body form? And this is in fact the case. We use this is the staining of human HeLa cells with a probe specific for satellite III DNA present on human chromosome 9, and the same cells are co-stained with antibody against HAP, and there is a perfect colocalization. If you use this a probe alpha satellite sequence specific for chromosome X, so it's still heterochromatin, but on a different chromosome and a different type of repetitive DNA element, now there is no colocalization. So there is a colocalization with a repetitive DNA element that is present mainly on chromosome 9, but also on a number of different human chromosomes. And of course, satellite sequences and heterochromatin are specific. Usually, you imagine there are specific heterochromatin markers that are found in this region. And one, for instance, is the methylation of histone H3 on lysine 9. And we tested if this marker is present in nuclear stress bodies, and is not. And the same is true for heterochromatin protein 1. Heterochromatin protein 1 should label heterochromatin portion of the region, constitutive heterochromatic portion of the genome, and is not present in these regions. So it should be heterochromatin, but it's not heterochromatin. And so if it's not heterochromatin, it's possible that it's euchromatin. And what we did, we used antibody against hyperacetylated histone H4, and you can see that this region where HAP is recruited after its shock and is composed of satellite III DNA, is actually decorated by antibody against histone H4. So is after its shock, this heterochromatic portion of the human genome becomes euchromatic, and as a consequence, we look at the presence of RNA encoded by this region, because heterochromatic DNA should not, I mean, at the time when we did this kind of experiment, it was supposed that constitutive heterochromatin should be silent throughout the cell cycle, should not be transcribed. And so we performed northern blot analysis with the RNA extracted from normal cells, not its shock cells. And we probed this RNA with a probe specific for satellite III DNA. And you can see here, no signal. Actually, by RT-PCR, now we know that there is some signal, but by northern blot, you cannot detect it. And at 42 degrees, after 1 hour at 42 degrees, you can start to see some signal. But then there is a massive production of satellite III RNA during the recovery from its shock. And then the RNA degrades and is reduced after a while. So which strand is transcribed? Actually, we found that only one strand, mainly one strand is transcribed. The satellite III sequence is composed mainly of G and A. So one strand is G-rich and the other strand is C-rich. You can probe with biadenylated oligonucleotide and see that it's actually the G-rich strand that is transcribed, that is present in stress bodies and colocalizes with up. So the RNA is sitting there, it's present within the stress bodies. And only one strand is present. And then you can decide to see if you can get rid of this RNA, and if this RNA is acquired for the formation of stress body, and you can treat the cells, for instance, with an antisense RNA, here antisense RNA or sense RNA in the sense compared to the transcribed strand of the satellite 3 sequence. And you can do also with siRNA against the satellite 3 RNA, and you can see there is a drastic reduction of the RNA present in this region. And you can see also the distribution of HNAP up and splicing factor SRSF1. The accumulation of these two protein nuclear stress body is drastically affected when you get rid of the satellite 3 sequences, but if you consider H factor 1 or hyperacetylated histone H4, these are not affected. So the idea, and this is the last for this part, I mean, these satellite 3 RNA are nuclear. They cannot be detected in the nucleoplasm. And we proved that either by northern blot analysis or by the typical heterokaryon assay that was used to assay the movement of the protein from the nucleus to the cytoplasm. I mean, if you have, you can distinguish here, for instance, a mouse cell nucleus because I have these dots, heterochromatic dots, and the human cell nuclei. And you can see that satellite 3 RNA are present in human cell nuclei, but they cannot move to the mouse nucleus. So they are confined to the cell nucleus. And so the idea that the model we have proposed at that time was that actually what happens is that you have long arrays of satellite 3 sequences in the human genome, and when you apply shock, what happens is that shock factor 1 binds to sequences present in the satellite 3 DNA, and recruits histone acetylases, and so you have a massive transcription of these sequences with the recruitment of RNA binding proteins that recognize that particular sequence, or they are recruited through protein-protein interactions. And then, and all this part was actually proven by all the experiments we and others have performed. But then you have progressive degradation of these RNA, and so protein are again released in the cell nucleoplasm, and you assume that eventually the cell return, the chromatin return to the initial status. So is it shock the only stress that can induce this rearrangement? And this is not. I mean, there are a number of stressing conditions that can induce the formation of stress bodies, and for instance, MMS, that is DNA damaging agent, etoposide, or cadmium, treatment with cadmium, or a low concentration, short treatment with cadmium, or a constant treatment with cadmium, UV radiation, hyperosmotic stress, but some other stressing treatment cannot induce. And as you can see here, for instance, by real-time PCR, now we can measure the abundance of this RNA molecule in the cells. And for instance, when you treat the cells with cadmium, you can have an induction of 60,000-fold, 100,000-fold compared to a basal level that is still detectable in the cells. But other, for instance, hyperosmotic stress, you have only around 1,000-fold induction, and so different stressing conditions induce the transcription of satellite 3 sequences with different efficiency and with different kinetics. And particularly in the case of, so the idea is always shock factor 1 that induces this kind of rearrangement or not. And I mean, if you treat the cells, each shock HeLa cells, and then you get rid of shock factor 1, you can see that now you have no longer stress bodies. But if you treat the cells with, you submit the cells to hyperosmotic stress, and you target each of factor 1, still stress bodies are present in the cells. And so one possibility is that there is another transcription factor can have exactly the same effect, and this is TON-ABP, the tonicity E binding protein that activates all the genes that are regulated by hyperosmotic stress, and in fact, tonicity BP binding protein is able to control the production of stress bodies after hyperosmotic stress. So what about the kinetics of this RNA? As you can see here, by our TPCR, we can measure the abundance of this RNA in the cells, and there is a basal level of this RNA in unstressed cells, but after it's shocked, there is a massive production of this molecule, and then the level of this RNA decreases, but still after 96 hours after its shock, you can still detect a level that is 10-fold higher than that detected after in unstressed cells. So you can distinguish different moments, an initial phase between 0 and 2 and 6 hours during which satellite III RNA molecules are synthesized, a second phase in which most of these molecules are degraded, because you can see here after 24 hours, the level of satellite III RNA molecule is reduced to approximately 1%, and then these molecules survive up to 4 days after its shock. And you can detect them also by in situ RNA fish. You can see here after 2 hours, 24 hours, but I don't know if you can see here, but there are 2 dots here after 72 hours. So you have a progressive reduction in the size and the number of stress bodies, but they survive for a very long time period. And so the idea was, can they be processed into small RNA? And we looked carefully to this possibility, and eventually we use LNA oligos, and we found that there are small RNA that are produced here. You can see a ladder of molecules from 25 to 75 nucleotides, with a spacer of 5 nucleotides that is exactly the size of the satellite III sequence. The monomer of the satellite III sequence is 5 nucleotides, and you can see there is a ladder of these RNA that are detectable essentially between 30 hours to 72 hours during the recovery from its shock. And they are RNA molecule because they are sensitive to RNAs, but they are insensitive to DNase treatment. They can generate it also in hamster cell lines when you supply the human chromosome, but they are not detectable in hamster cell lines without the human chromosome. And the kinetics is very similar to that observed in human cells. These small molecules derive from pre-existing molecules. So the idea is that they derive from the processing of the large molecules that are synthesized at the initial phase of the response to its shock. And in fact, if you inhibit transcription with actinomycin D, now for instance, you have a reduction of the level of CMYC because it's no longer transcribed, but you still have the production of these small RNA molecule, satellite III RNA molecules. And they are mainly found in the nucleoplasm. They are, some of them can be detected in the nuclear pellet. Some of them are detectable in the nucleoplasm, and eventually very, very little in the cell cytoplasm. So what happens is there any effect of its shock on cell cycle progression? And there is some link between the production of this RNA molecule and cell cycle progression. And what happens is that if you shock the cells and you measure the fraction of cells in S phase, or in G2 phase, or in G2M, or in this is S phase, G1, and this is G1, S phase, and G2M. So you can see that there is a change in the relative abundance of these three phases in the cell, of the cell cycle. And between 12 and 24 hours, there is accumulation of the cells in G2M. And what happens is that you have accumulation of the cells in G2M, and then progressively the situation returned to the initial status. So this is similar to what we observe here, because there is accumulation of this RNA before 12 hours, and then there is this intermediate phase, and then cells return to their original status. So what happens when cells accumulate in G2M? When cells accumulate in G2M, you can see here, most of the satellite 3 sequences are lost. Are lost because they are no longer associated with chromatin. They are no longer associated with DNA, but they can be found in the cell cytoplasm. And if you look at cells in anaphase or in telophase, you can see that these molecules that are stained in red here are in the space between the two chromosomal sets. So why they are present in this space between the two chromosomal sets? Why they accumulate in this particular region? So the reason is just because they are associated with chromosomes. Its shock induces a change, it affects the migration of specific chromosome during mitosis. So there is a specific set of chromosomes here. If you look at cells after its shock, you can see there are chromosomal bridges or delayed chromosomes. And the abundance of this particular structure during mitosis peaks between 6 and 12 hours. And what happens is that satellite III RNA is associated with delayed chromosome and chromosomal bridges. And if you decorate the cells with a probe specific for satellite III DNA or for chromosome 9, you can see that delayed chromosome and chromosomal bridges are decorated by satellite III sequences. So the idea, the idea that we have now is that, so this body structure formed in mitosis contain RNA binding protein. Here is a splicing factor SRSF1. Here is HNAPM. And here is the distribution, is the electron microscopy analysis of this structure in mitotic cells. You can see this is a chromosome, and here you can see a stress body. You can see a chromatin bridge that connects the chromosome with the stress body, or again, a chromosome, a stress body, again here, two chromosomes and the stress bodies. So you can see that even in mitosis, this structure persists and they are associated with chromosomes. But what happens is that here, you can see there is a delayed chromosome here or here, and you can see there are two stress bodies associated with this delayed chromosome, and the structure and the form of this delayed chromosome is highly suggestive of the presence of an entire chromosome that is not undergoing sister chromatin separation during mitosis. So the idea is that actually what happens is that you have activation of satellite III DNA sequences that are close to the centromere, and in this way you can alter the the functionality of the centromere and perturb the migration of specific set of chromosomes during mitosis, and first of all of chromosome 9. And I will stop here. Thank you.